So I'm going to get started. Welcome to the webinar. My name is Hannah Brackey. I am the Director of Product Management here at LabKey. And today I'm going to talk about setting up your research lab for managing clinical trial samples. This is a subject I am fairly passionate about. I had spent several years in my previous life before LabKey working in a lab and starting a lab that was managing clinical trial samples. So we started out with the one patient getting maybe a sample every couple of weeks to by the time that I left running 5 consecutive trials with multiple samples every day and trying to coordinate that. So maybe just some general comments about the webinar in general. We're going to have a few polls today. As questions come around, feel free to add them in the chat. We'll definitely have a Q&A session at the end, but feel free to just type things in as we go, and I'll either answer them as I can or we'll answer them at the end. So for today's agenda, we're going to talk about 2 different things, both the regulatory considerations for managing clinical trial samples, as well as some tips to help maximize the efficiency and help your lab manage those samples as you continue to grow. So we're going to start out with some engagement and see if how comfortable you guys are feeling that your lab is following the necessary regulatory guidelines. So I'll try and open this up here. And so feel free to just answer those. We will circle back to that in a few minutes and kind of gauge people's comfort. Hopefully, most of you are feeling comfortable, but still want to learn something today. So I bucketed this out into really 4 major regulatory considerations, and these are all really going to be useful regardless of the regulatory agency or body that you guys are reporting to or wanting to follow. So the traceability of samples is really key. You're probably here because you know that that's an important aspect of being able to manage clinical trial samples. And the short version of this is really for any sample that you receive into the lab, any lab member or auditor, somebody coming in, should be able to answer what that sample is, when that sample might have gotten received into the lab, when it got stored, when it got processed, and then the how questions. How did those things happen? What were the procedures around that led to that result or led to that sample being generated? Secondly, sample identification. So making sure that you're able to create clear and unique sample identifiers that are appropriate to your lab type. So this really means, do your samples need to be de-identified, or can they continue to have PHI on them? Do you have varying levels of that? What type of identification pattern do you guys want to follow? We'll get into that in a couple slides as well. And then also having standard operating procedures or SOPs. So this is not always the most fun of topics, but I think it's really one of the most important. So really being able to create and generate SOPs that work for you, that are actually followed, and that do truly document all of the planned and really unplanned sample related tasks as well. Record keeping, which is similar to SOPs, but the reality is that as well as sort of following the standard operating procedures, you may also want to either include in your SOPs ways to document what you've done or have a separate record keeping system for that. So we've given it a couple minutes here. I will end the poll and we'll share the results here. So it looks like, yeah, a lot of people are feeling, fairly comfortable, which is wonderful. I think that's what everybody wants to see. But there's some small changes that can be done, and so I love to see that, and hopefully we can talk about those small changes today. So let's dig into the traceability of samples a little bit more. Those what questions. So something to consider is documenting the types of samples that you plan to both receive into the lab as well as create. And so if you're receiving a bunch of tissue slides, are you going to be slicing those up and storing them in different fixatives? Are you getting blood and then creating PBMCs or DNA samples, things like that, making sure that you really know what those samples are, and documenting that, as well as considering sort of the source of that sample. So in many cases for folks, this is going to be a participant source, somebody who is enrolled in the trial, as well as the derivatives of those samples. So as I mentioned before, if you have a blood sample that you're driving DNA from, you want to ensure that you know that that DNA came from that blood sample that came from that participant. And then you also want to make sure that you're going to include pertinent data that you want to collect for each sample. So this type of data is really information that's going to help you in the laboratory, as well as help you with an audit. So you want to consider what the lab needs, as well as sort of what an auditor is going to want to know. And that's typically these when questions. So as part of this data collection, making sure that you are capturing these when fields. When was the sample collected? When was it received into the lab? When was it processed or stored? And then you can even kind of advance these into how long was the incubation time? So you can evaluate if that, that sample or data integrity has been lost along the way. How you did all of this, as I mentioned, all of the different steps that you would take in your sample receipt, that you're checking those tubes for breakages or cracks, possible contamination, the fact that maybe it hadn't been stored at the right temperature, all of those things should be documented and easy for an auditor to see. And then this isn't as commonly stated in by regulatory bodies, but really the who of all of this as well. So it's very, very good to know who did all of these things. And that's good both for an auditor, but then also for troubleshooting in your lab. If you need to explore something, who was the last person that grabbed that out of the freezer or did that processing task because the data might have looked a little bit strange. And then for sample identification, really consider that initial receipt of samples if you are receiving them into your lab. So how are those going to be labeled from the collection site? And do they need to be de-identified, as well as sort of sample accessioning? Once you've identified that, you can really start to determine the laboratory specific identification. And there's, a handful of layers to this. So I mentioned before, do you need to de-identify, or maybe not need to de-identify, but do you want to de-identify those samples for security purposes? As well as considering that sample uniqueness. So a lot of people will use barcodes to uniquely identify a sample. You can also create human readable sample identifiers with that just have uniqueness constraints. So you're able to read a label on a tube and know exactly what's in it rather than needing that barcode scanner. If you're going to go that route, it's very helpful to define a systematic naming pattern. So human readable is always desired. This can be very useful when you're in a lab and there's samples on the bench and somebody comes up and reads that sample. They're going to know exactly what it is and what maybe precautions they need to take as well. When you're defining this naming pattern, also account for any sample aliquots, any derivatives of that sample that you might be taking, and then also repeat samples. So this happens, a fair number of times for clinical trials where the sample gets broken or hopefully not lost in shipping, but certainly that has happened to me before, or the incubation time is too long. And so a repeat sample needs to be taken. And then think about how that naming will scale over time. So if you're using auto-incrementing IDs, for example. Do you want to have any leading zeros ahead of that to kind of make consistent naming? Is that an important aspect of what you're doing, or just a nice to have? And then the last one is sort of this reality in the lab, right? So you can go through all of this and create a great human readable naming pattern. You can have a 16-digit barcode that's going to be super unique. And then none of that is going to fit on a label. So how are you labeling and identifying samples that is truly going to fit in that 1 millimeter by like 50 millimeter label? And then will those samples be easy to identify? So again, either, are barcode scanners very prominent in your lab? Maybe if you're barcoding everything, it's very easy. People are able to identify that very quickly. Or if you have those human readable identifiers, that will be a lot easier as well. And so for SOP standardization, excuse me, we've got a couple slides here. And I think, we're coming from the point of perhaps you guys have some SOPs. Maybe you have, that you need SOPs, but they're not quite in place, or maybe your SOPs are outdated. So the first thing that you really want to do is kind of draft that sample life cycle of what's going to happen to those samples within the lab. That really includes anything from receipt to that whole sort of processing, storage, any derivatives or experimental data that you'll be collecting on that in your lab. Making sure that you also identify areas of possible deviation. So what happens when you expect 10 mils of blood into the lab, but you only get 2? What are the procedures that you have in place, and create the touch points around that? Also, make sure that you're identifying key variables about the sample during this life cycle. So things that might be important are those incubation times, media diluents, the temperature of thawing or incubation or storage, things like that. And then also making sure to capture decision points. So if you expected to get a certain number of cells out, and it's quite a bit lower than that, what are the decision points around that? Do you have those SOPs in place as well? And then once you've done that, really crowdsource this to your team and make sure that the team that's performing the work also agrees with you, because there's no point in creating SOPs that nobody is following. And so you want to really engage your team members and make sure that they're on board with what is being written. After that, either create or update your SOPs, or sort of a mixture of both. Compare that sample life cycle with your SOP library and check for 2 major things. So do the SOPs that you have capture that full life cycle? Are there gaps? Are there things that are missing that you want to create or update SOPs for? And then are those SOPs up to date? So these really should be less static documents and definitely version controlled, but making sure that they are getting updated as things change in the lab in sort of that regulated way. After you've done this, if you have time, I would always recommend doing sort of a trial run of the sample intake, processing, and storage. After that trial run, you can then sort of complete a practice audit. If you don't have time for a trial run, I would still complete a practice audit on the samples that you've already received. And make sure that you can identify those 4 major things: the what, the when, the how, and the who, and kind of always dig in a question further to make sure that it's really being answered to the fullest. And then, of course, capture those weaknesses in your documentation or process and make improvements to make sure that those are being captured accurately in your SOPs. And then the biggest thing of this is maintaining it, right? So you want to make sure that you have an SOP for when you want to review your SOPs, and also how change processes are going to get put into place as well. And make sure that you're fostering that environment where if something isn't working for team members because it's not scalable, it is not working maybe with the instrumentation that you have, really having people come to you and be able to say, hey, this isn't working. Are we able to change this? And then making sure that that gets documented. So definitely having an SOP for SOP change requests is going to be important. And then also continue with those regular internal audits. The best way to be sort of prepared for an external audit is to have those on a regular basis, to make sure that things are running in the lab smoothly. And then that record keeping component. So really developing a system for tracking that what, when, how, and who. This could be through an electronic lab notebook. It could be through a sample management system. It could be on paper. It could be, in Excel, although that's not always desired. But really making sure that everybody is on board with how that documentation is both stored and shared in the lab. So if you've got paper SOPs, having that master SOP binder in very easy to access locations as people are doing their work, or a file on a shared drive, something like that. And then also make sure that you're considering those regulatory requirements around record retention. So most bodies are going to require you to keep it for a certain amount of time, and that kind of varies between the agencies. So look into that and then kind of come up with a plan for making sure that you're retaining those records. And then, of course, like staying organized. So even, paper is an easy one to point to, but even electronic records can really get very messy very quickly. And so if you establish an organization system or have a tool for this early on, it's going to allow for that easier data recall during an audit. So we're going to shift gears a little bit and start talking about the efficiency and growth tips that we have. So I'd also like to start out by asking you how you guys are feeling with your lab's efficiency. So let me get this poll started. And we'll let you guys answer that. And again, we'll kind of come back to those results in a slide or two. So my first tip for maintaining and establishing lab efficiency and being able to grow is making sure that you're flexible, but in a very smart way. So this is a regulatory environment. You want to make sure that things aren't changing on a daily basis or even probably on a weekly basis, but you do want to make sure that you are adapting to the new workflows that come based on maybe new sample types or new requests that have come in from the trial staff, as well as challenges you might be facing in a growing lab. So back to those SOPs, making sure that you're developing methods to change and adapt those standard processes as you need to. And that's really going to be vital in tracking the changes that are happening in the lab. Making sure you're evaluating often. So looking at these processes and resources, making sure that you have enough staff for what's coming, that you have enough instrumentation, that you have enough reagent on hand, that you have a good system that's going to help you track all of these things as well in an efficient way is going to be important. And then also making sure to, continue to engage that team, right? So one of the keys to efficiency is people being satisfied and happy in their work environment, and that, they're not facing a lot of barriers as well. So there's really an emotional component to this where you want to really foster that culture of continuous improvement. And then, of course, use a sample management system that supports those flexible working environments, but also is going to maintain that reliable data capture that's going to be needed for an audit. And then another thing to do is really to help plan those data requirements to make sure that you're being as efficient as possible from the beginning. So going back to that original sort of sample life cycle mapping, make sure that at any point in that life cycle, you're wanting to capture all of the information around those samples that it's going to be helpful for your lab, as well as what is going to be helpful for an auditor, so that those what, when, how, who questions, and making it really relevant to you. So that's kind of that first point. Keep it as simple as you can. So if you're able to combine similar data and labeling that appropriately, that's going to maximize efficiency in the lab. If you've got 50 different fields that only get filled out sometimes for people, it's going to take folks a lot longer to enter that data in than it would be if it's a couple of required fields every time. And then, of course, creating those templates. So in order to make things efficient in the lab, you can create templates either electronically or on paper that make that data entry really easy. You always want your scientists to focus on the science and not necessarily the logistics of how to capture data around what they're doing. So making that as straightforward and simple as possible is going to be important. And then also making sure that your freezers are audit ready. So I have been in labs before where we have varying levels of freezer management, both from everybody gets their own shelf and it's kind of a free-for-all, to having a very structured, organized system. So this will obviously change as the lab continues to grow and collect more samples. Other things to consider around not only record retention, but also sample retention. How long are those going to need to be around for? Because that will likely scale the amount of storage that you need as well. So as you're doing this, or as you're establishing these systems, really make sure that you're keeping naming consistent. So scaling to more freezers will be easier if you're all talking about shelves and racks and boxes in the same way. Also establishing guidelines on how those samples should be stored. So there's this sort of ever-long debate of, do you want to maximize space to the fullest? So anytime there's an open spot in a box, samples are going into that. Or are you wanting to compartmentalize? So maybe there's DNA on a certain shelf and RNA on a different shelf because you're accessing them for different assays later on. Or do you do it by participant? So if somebody revokes consent, it's a lot easier to grab those samples. And then make sure that you're capturing that storage accurately in something like a sample management system. So the reality of this is you always want to make sure that you're prepared to identify where a sample is being stored, and of course, under which conditions. So the last thing here that I'll say is really do your research on sample management systems. There are a lot of good ones out there that will help support this type of work. It makes your life easier, it makes auditors' lives easier, and truly you do kind of see the value in these over time as the data entry becomes very consistent. Choosing that system can really have a lasting impact. I think I recognize some names on the participant list here. We all know that migration and change is hard. And so really making sure that you're finding a solution that works for you and your team in the long term is a good idea. So look for those must-have features. So determine and document those. Write those up, talk to your lab, make sure that you're following sort of the regulatory agencies that you're being audited by, all of their rules and guidelines on this as well. making sure that you're finding that tool that's going to allow for that flexibility and further growth of your lab as you continue to receive more samples. And then of course, like, get the team involved, right? Make sure that they're also evaluating it, demoing the products, and getting them really excited about it. Again, this, change management is very challenging. If people are at least like less than hating it, that's definitely going to be a good sign. Ideally, you would have them love the system that they're going to use, and that's what we're really aiming to do at LabKey. And so maybe before we start this next poll, I am going to end the efficiency one. We can take a look at it quickly. So it looks like most people are feeling kind of okay, but definitely need to get better at efficiency. And so hopefully some of those tips helped. I think other things to consider is really, scaling those systems as you can, and planning ahead again for the future, I think will make people feel more comfortable with that. So the last thing I'll ask of you guys here is to let me know what sample management solution your lab might be using right now. And as you answer that one, I will kind of go through LabKey Sample Manager. So Sample Manager is one of our newest products at LabKey, and it was really designed to bring efficiency and that ease of use to clinical sample management. So I'm seeing some of you have already entered that you're using commercial software. I've also used commercial software in the past. Not all of it's easy, and we want to make people excited in the lab about using it, also while fulfilling those regulatory requirements that are needed. So some things about Sample Manager that really help with this are our full history and chain of custody on each sample. So we have a sample timeline that tracks anything that's ever happened to that sample since it's been registered in the system. You also can customize all of the different types of metadata that you need in your lab to track on your samples, both for sort of that normal day-to-day lab work, as well as anything that you might want to track for an audit as well. We also support barcoding. So we both support, we'll create barcodes for you, but then if you also are coming in with your own barcodes, we allow for that as well. And then we have a very advanced either auto or manual mechanism to create unique human readable sample IDs, which can be very helpful if you don't just want to have a barcode on your samples. And then we also enable for you to create templated task-based workflows that accurately reflect those SOPs. And so a lot of people will write their SOPs and want to follow that record keeping component and say, yeah, a workflow is going to work for me. I can capture that here. As well as making sure that you can retain the records of the sample even after it's left the lab, and maybe that's left the lab as in being shipped out, or left the lab as it's been consumed or discarded. Instead of having to traipse through an audit log, we allow for that record retention and pull up very quickly. And then, of course, having that integrated and auditable management of freezers and sample storage. So we're tracking all of the things that are happening with your sample, including that storage management component. There are 4 main feature areas of Sample Manager. So sample tracking, as we discussed, we're tracking that full chain of custody of the samples. You can create that metadata. You also can easily navigate that sample lineage. And so it's really easy to see where a sample came from, and then also be able to search for samples based on where they came from or where they were derived from. The freezer management component, so all of the things that you would expect out of a normal freezer management system, being able to match your digital storage with your physical storage, tracking the storage and movement of samples, being able to make comments on that as they're coming in and out of the freezer, why they're being used, things like that. And then of course, tracking the amount and the freeze thaw history is kind of automatic. Our workflow management tool really helps with that standardized SOP management, being able to track those lab processes. You can assign this work to different people in the lab. And so as a lab manager, you can kind of dole out work to other people, or as a member of the lab, you're also able to see the work that's been assigned to you and really focus on getting that done. And then last, we also have an assay data capture component. So really being able to see all of your system, all of your data in one system on your samples, and being able to track the status of those experiments, either via workflow or kind of on their own. And some things I'm really excited about coming up for Sample Manager. In the next couple of months, we're going to incorporate the LabKey Biologics electronic laboratory notebook into this product. So you'll be able to track and manage your samples in this very flexible regulatory environment, as well as capturing your electronic lab notebook here. And so this really allows you to document those experiments and highlight the analysis and reporting that you've done. We do include these data linked documents by directly referencing any of the data that you have within the system. We'll also be adding some additional custom reporting, so creating visualizations to help you quickly evaluate and QC your data, as well as generating custom reports for either downstream analytics, or if you're reporting to regulatory agencies, those are going to be really helpful as well. And then some new quality control features too. So being able to set custom QC statuses and separate permissions for folks that'll be able to do that, as well as a specialized QC workflow task that really will be designed to sort of gatekeep requests and validate any work that's been performed. And so kind of lastly here, I do want to say the best way to learn about Sample Manager is to try it. So I think Will is putting in a link to a demo in the chat. I encourage you guys to click on that, schedule some time with me. I'd love to show you around. I'd love to talk to you about what you guys need and how we might be able to help you. So with that, I think we'll kind of leave it at the Q&A and see if folks have questions. Let's see if I can get to the chat here. Yeah, so there is a question, is LabKey Sample Manager cloud product? And it is. And so we offer this as in the cloud, and it's something that we host for you and do all of the maintenance and management for. And so it's very easy to implement and set up, and then, have us maintain over time. Yeah, so there's another question here. If somebody needs a local installation, we do have offerings around that if it needs to be on premise. Primarily, we like to refer folks to our larger product suite, LabKey Server, for this as well. But definitely, if you're interested in what Sample Manager has to offer, set up a demo and let's talk about it. Yeah, so another question coming into is the ability to sign off on work done within Sample Manager. So this is really where that new quality control functionality is going to come through, where you can set permissions for people to QC the data and have that specific e-signature around it to validate what's happening in the system. So that's coming soon. Yeah, and somebody is talking about migration as well. And so we definitely support migration from other commercial systems, from Excel. We haven't yet done like a full-on paper migration yet, but usually there's something electronic for us to use. But we definitely support you every step of the way. It is very easy to get data within Sample Manager. If you set up a demo, I'd be happy to show you. But we always will dedicate a person to help guide you on what the best structure is to really help you succeed, because that's the ultimate goal for us. And is there a limited amount to data storage? So we don't actually have set limits right now, although I think it's kind of sort of this within reason thing. So definitely, if you are looking to store genomics data or other sort of very large data types, set up a meeting and let's talk about it. Generally, we'll come up with something that works for everyone. Yeah, so somebody is wondering if LabKey is a really new company, and how does this look like for a group that's really wanting to scale over time. So LabKey has been around for 19 years. We are a scientific data management company, so we are skilled at this. We've been doing this for a long time. Sample Manager just happens to be our newest product that's a little over 2 years old now. And so we really are in it for the long haul here. We also do provide, in the case of some catastrophic event, there's always the ability to fully export and get all of the data that you have in your system. So that's never going to be lost. It's never going to go into a black hole. It's something that you always own and control. Yeah, so somebody's asking about a practice audit process. So what I generally have done in my past life is to identify a set of pieces of information that I want to do in my audit ahead of time. I usually will work with somebody, typically outside my lab if I can, of these are the things that I want to ask. So I'm making sure I'm not biasing myself on, oh yeah, I know that we're really good at receipts, so let me only audit receipt. Let me kind of get that full gamut. But generally some good practices are to sort of ask questions of, if I were to pick any sample, what is the information that I'm going to be able to find out about that sample? Where is it currently? Who is in possession of it currently? And what is the information or things that have happened to it since to kind of get to that point? And if you're able to pick a sample at random and be able to identify that, I think you're already in really good shape. Some other things to identify is taking a look at a random SOP and seeing the documentation and record keeping around that SOP, and see if people are deviating from that, or if it's on track, or if there are deviations, are those being recorded and traced? So those are some general good practices. It doesn't have to be extensive, right? You don't have to prepare a full 2-day audit. I would say do something that's going to take an hour or two every 3 or 4 months, and that should at least give you a good idea of how your lab's doing in terms of SOP management. And yes, somebody is asking about HIPAA compliance. So Sample Manager is HIPAA compliant at the professional level, and that's something that we do offer.